Today, I'm going to show you transcription. Transcription is the first part of how a cell makes proteins. It's called protein synthesis. This is because the DNA cannot leave the nucleus, so it has to send out a messenger in order to make the protein. So there's two parts to protein synthesis. The first part is called transcription, and the next part is called translation. But for our purpose today, I'm just going to show you by demonstration transcription. So, you know that we had DNA nucleotides, but we also have RNA nucleotides. I'm going to show you the difference between a DNA nucleotide and an RNA nucleotide, both of which are made in the nucleus. Take out two. These are two nucleotides. This one is a DNA nucleotide. This one's an RNA nucleotide. You can see that they both have three parts: phosphate, sugar, and a base. However, there are two differences that you see here. One difference is the sugar is different. In a DNA, the sugar is called deoxyribose. That's where the DNA gets its name from, the sugar, the D. You can see it's a straight back over here the way I designed it. And in an RNA, the sugar is the lighter blue, is called ribose. That's where RNA gets its name. The phosphates are the same. So the sugar is the big difference between them. Also, we notice that there are 4 different bases in DNA: A, G, C, T, and in RNA it's the same. We have A, G, and we have C, but instead of T, you can see these two are different. There is no thymine. This is adenine, this is cytosine, this is guanine, this one's thymine, this one is uracil. So one of the bases is different. It's called uracil. Uracil is a single ring base, so it's a purine. Alright, just like thymine is. So the base pairs would be the same. A would always base pair with T, and just like in the RNA, the U would base pair with A. So U takes place of where T would be. That's the only difference in how we base pair. So it's AUGC for RNA to DNA. Okay, so let us now start transcription. First, the purpose of transcription again is to make an RNA copy of DNA. So what I'm going to do is set up a ladder with DNA. I'll make it 6-6, a 6-step ladder with DNA. How's that sound? Okay. So then I'm going to base pair. We got C and G, C and G, T and A and T, G and C, and T and A right here. This all occurs in a nucleus. The DNA never leaves the nucleus. Okay? The same thing happens. We're using very similar words. We have an enzyme. My hands are enzymes. That's going to break the weak hydrogen bonds between the bases. That enzyme is called helicase. You remember helicase from replication. So here we go. Helicase breaks the bonds between the DNA. Okay? But we're not replicating. What we're doing is we're only copying one side. So we're going to pick a side. Let's pick this side. So I'm going to take my RNA nucleotides and base pair with only one side. One of the big differences between DNA and RNA is that RNA is only single-stranded, unlike DNA, which is double-stranded. So here we go. I'm base pairing U with A, G with C, C with G, U with A with T, and G with C. And there are two differences. Instead of T, you have U for RNA. So now this comes together by another enzyme called RNA polymerase. So this is together. This RNA polymerase, then this RNA splits off and leaves the nucleus, this strand that we made right here is called mRNA. This is the messenger. Made a copy of one side of DNA. It's a single-sided copy of DNA. And this messenger is going to leave the nucleus and go to the ribosome, where it's going to direct and continue to make proteins. And a protein is a command from the DNA. So this is going to leave, and then DNA polymerase is going to put the DNA back together. So it only temporarily unzips. So RNA can come in and copy one side. This new strand we made is called mRNA, messenger RNA strand. And then the DNA zips back together again. So it's very similar to replication, but we made this. This is what we wanted. This is our mRNA strand. This is what we're looking for. This strand goes from the nucleus to the ribosome. This is the first step of protein synthesis called transcription.